Okay, now this is Miss Cargan, and we're continuing our steps of protein synthesis. And we're taking a look at how the mRNA goes through the pores of the nucleus, and that way it's copying the DNA code and taking that code out of the nucleus and into the cytoplasm where you find the ribosomes. And that's where the actual protein synthesis takes place. So this is what happens at the ribosome. So this is that second step in that central dogma that we talked about earlier. So this is the process of making a protein translation. And this is the second step. This is where at the ribosome, so you see the ribosome here, and remember it has two subparts. It's got a large subpart and then a smaller subunit. And then this is the decoding of the mRNA. So this mRNA, which came from the nucleus and is now in the ribosome, and it is being decoded. And by decoded, we mean the ribosome is bringing tRNA pieces, and that's what we see here, these kind of T shapes. This tRNA is bringing amino acids. You'll notice right here this amino acid lysine is being brought in, and these tRNAs are bringing in based on the codons here in the mRNA. And each tRNA has what is called an anti-codon, which matches with these codons. So this process begins with the transfer RNA bringing amino acids from the cytoplasm to the ribosome. So again, this step is called translation, and it is where the transfer RNA carries amino acids to the ribosome, and that's where it hooks up with this mRNA. So you have mRNA and tRNA, and that tRNA is bringing amino acids to the ribosome. So I will go on to the next step, and again, if you need at any point to stop or rewind or pause the video to get the notes, please feel free to do so. Now, one of the biggest questions that I get asked is, where do these amino acids come from? Because there's just amino acids in the cytoplasm. And these amino acids come from the food that we eat. The food that we eat that contains protein, our body takes those proteins and breaks them down into individual amino acids, and then we rearrange them. For this, I like to think of, say, Lego blocks. Like you can have Legos that are built into any number of shapes, and then if you want to reuse those Legos, it's easy enough to break down a toy that you've made and make it into something else. And that's basically what our body is doing here, is they're breaking apart those amino acid Lego blocks, and they're using those blocks to create something new based on the directions that they're getting from the DNA. Now, anytime on mRNA you see three base pairs, again, here you have guanine and cytosine and adenine. Anytime you have three base pairs that are side by side, we call those a codon. And that simply means a little piece of code. So this is a little piece of code. It's 3 bases long. And this is only for mRNA. This codon is for mRNA. Now, in tRNA, you have what is called a complementary codon or an anti-codon. So this codon would have G, C, and A, and the anti-codon for G, C, and A would, of course, be C, G, and U. We'll talk about why it's U and not T. Because, of course, this is RNA. We're not talking about DNA at this point. So the triplet of nucleotides in tRNA is an anti-codon. So what is a codon in mRNA becomes an anti-codon in tRNA. So this is a tRNA. You'll notice that it is attached to an amino acid and that it contains this series called the anti-codon. Okay. Each tRNA is going to code for a different amino acid. So whatever this anticodon is, it is going to attach to one of the 20 different amino acids. So each one of these codons holds the key or the code for a specific amino acid. Okay? So mRNA is carrying these instructions from the DNA, which of course is located in the nucleus. So this mRNA comes out of the nucleus carrying this code, okay? And in the ribosomes, these tRNAs bring the amino acids, okay, to the ribosome, okay? The ribosome positions itself, and there is always a codon called the start codon. And this is what starts this whole process. This start codon starts with an amino acid called methionine, and that means that it's going to begin this process. Okay? So as you bring in these tRNAs to the ribosome, you have an amino acid chain that begins to form. You'll notice here this chain beginning to form. Okay? We call this a polypeptide, and we'll use this term again later, but basically what I want you to know is that these amino acids are joining together to make a protein, okay? Polypeptide is simply another word for protein. Okay, and now we're going to practice using a codon chart. And we'll come back to this slide here in a minute, but I want to show you what a codon chart looks like. Now there's two different kinds of codon charts that we're going to use in our practice for this. This is the first codon chart, and what this is reading is you are reading the mRNA. So when you have mRNA, because again, mRNA is the only type of nucleic acid that has codons, okay, your first base is going to be down the left-hand side. Your second base will be on the top here, and the third base will be down the right-hand side. So we are going to try to decode the CAC codon. So what we're going to do is decode CAC. So we look at the first base, which is a C. Okay, so we are in this row right here. So we know that we have to be somewhere in this row because our first base is a C. Then our second letter is an A. So our first base is a C, we're in this row. Okay, if our second base is an A, we're in this column. So we know that we have to be in this square, and you'll notice there's a CAU, a CAC, a CAA, and a CAG. So here in CAC, again, your third base, you can look at it here, CAC. So CAC would be histidine. So that first codon here, the CAC, is going to be histidine. Right? Now I'm going to talk you through this again in case you didn't follow the way we read this chart. Okay, so our second codon is CCA. So the first base here would be C. Okay, so we're again in this row of amino acids, and the second base is C. Okay, so we're in this block of amino acids, and here our third base is A, so we have CCA is proline, so that would be for our second. So what we're doing here is we're taking a look at this mRNA code, these codons, and we're looking here, and this codon chart is actually telling us what amino acids, and you'll notice there's 20 different amino acids. What amino acids do these strands of mRNA code for? Does that make sense? So what I'm going to do is I'd like for you to pause the video here, and I would like for you to try to use this codon chart to decode this UGG and UGA. So if you could do me a favor and pause the video at this point, I'm going to give you some time to try to read this codon chart and tell me what 2 amino acids you would put in these 2 blanks. So pause the video here and we'll come back to it and I'll give you the answers. Okay, now that you have completed this, hopefully paused the video, worked through decoding the codons UGG. So we have here the first base is U, and the second base is G. Right? So we're in this UG, and the third base is G. So this third codon would be tryptophan. Okay, tryptophan. And then the fourth of these we have U. So we're in this top row, and G. This in this last column. So UGA would be the stop codon, which of course would tell us to stop the decoding process. So this particular protein would have a histidine amino acid, a proline amino acid, a tryptophan amino acid, and it would stop. Okay. Now I wanted to show you another option. We do not know, we actually never know what kind of codon chart we're going to see on the standardized tests here in the state of Texas. So I want to teach you also how to read this one. Now, many biologists and geneticists prefer this codon chart because it's, in my opinion, much easier to read. And for this one, you simply start in the center. So you have an A, a G, U, C, a G, U, A, and C. And this is the first letter of your codon. So we have a letter A, so we're going to start here in this quadrant. Then you go to your second letter next. So we start here with A, then we go to G, because we go to U, so A, U, G. So we're going to go A, then U, okay, then G as our third letter. And that, of course, is methionine. This is our start codon. So this would be methionine in the first blank. The next is AAC. Okay, so you would start with A, and then you would go to A. Okay. Then you would go to C, which would lead you to asparagine. Okay. So again, I'm going to stop so that you can pause the video and practice. I would like for you to try to use this codon chart to decode GAC and UAA. So GAC and UAA. Okay, hopefully you've had enough time to get those two done. Of course, if you start here at G and A and C, you should have come up with aspartic acid for your third codon. And here for U, A, and A, of course, is our stop codon, telling us to stop that decoding process. So this is a little bit about how to use a codon chart, and this is to predict the amino acids that would be created by the mRNA. So I'm very sorry for the glitch. I had a problem with the PowerPoint, but I want to wrap things up here with the protein synthesis. This is again our central dogma, and of course we started with the DNA. This is in the nucleus, and the process of transcription, which makes a copy of the DNA to mRNA. and mRNA, which becomes translated into a protein. And this was the process we were doing with that codon chart, which takes place in the ribosome. So now that we've wrapped this up, we have a codon activity, and I have some instructions in a secondary video for how to complete that codon activity.